Hi, today we're going to talk about alcohol dehydrogenase. Alcohol dehydrogenase is an enzyme within the body which breaks down ethanol. Most of these enzymes can be found within our liver or our lining of the stomach.
When a person is having difficulty handling alcohol, they tend to develop an increase in body temperature and an increase in heart rate, followed by a reddening on the face and on the neck. Here's an example. This is known as an Asian flush. It is more common in Asian descendant because they lack the genetic code of ALDH2 alleles. Alcohol dehydrogenase 2 gene is responsible in the regulation and production of alcohol dehydrogenase enzyme in the liver. ALDH2 allele is dominant. This means that if a single cell copy interferes with a fully functioning formation of ALDH2 enzyme, it will result in an increase in alcohol buildup in the body. This means that acetaldehyde cannot be broken down.
The first step of ethanol metabolism is the oxidation of ethanol to acetaldehyde catalyzed by alcohol dehydrogenase containing the coenzyme of NAD+. The acetaldehyde is further oxidized to acetic acid and finally CO2 and water through the citric acid cycle, which is a Krebs cycle. A number of metabolic effects from the alcohol are directly linked to the production of excess both NADH and acetaldehyde, which is there. In a healthy non-alcoholic drinker, there are 4 different enzymes which takes part in metabolism of ethanol. We've got the alcohol dehydrogenase, acetyl dehydrogenase, cytochromes, and catalase. They are mainly found within the liver and catalyze ethanol by oxidation. However, in adults who drink alcohol at moderate level, even just occasionally, the ethanol is oxidized by acetyl only by the class 1 of alcohol dehydrogenase. This oxidation occurs in the cytoplasm of the hepatocyte, which is a liver cell, and this reaction is reversible. This means that the fewer enzyme will be needed for those people who drink than those people who don't drink. The major effects with the alcohol does to the body are in the liver. For chronic alcohol user, the alcohol can inhibit the major pathway of the fatty acid metabolism. Because the alcohol is just full of calories and no nutrients whatsoever, a fatty liver, which is an accumulation of fat within the liver cell, will then be delivered, which then causes inflammation of the liver.
Ethanol effects on mitochondrial function. The mitochondria are an important component in the alcohol metabolism by the enzyme aldehyde dehydrogenase, ALDH. This enzyme helps catalyze the conversion of acetaldehyde into acetate. When the substrate acetaldehyde concentration reaches too high and where the acetaldehyde dehydrogenase enzyme are fully saturated, the accumulation of acetaldehyde then leaks into the bloodstream and will result in damaging biomolecules such as lipids, proteins, and nucleic acid, leading to toxic side effects. This will eventually inhibit the mitochondrial reaction and functions. Within the mitochondria, aldehyde dehydrogenase 2 metabolizes acetaldehyde to acetate. When this enzyme fails, acetaldehyde builds up and damages the electron transport complexes 1 to complexes 4, causing a rise in production of reactive oxygen species affecting the electron transport chain and oxidative phosphorylation, disturbing the ATP synthesis. From what we found out, there's a second alcohol metabolic pathways for long-term alcohol consumers. This pathway is called the microsomal ethanol oxidizing system, abbreviated as MEOS, which takes place in the smooth endoplasmic reticulum within the liver cell. MEOS pathway via the cytochrome P450 helps convert alcohol into acetic aldehyde. This pathway requires oxygen and NADPH to convert NADP and H2O. This enzyme carries a trait which has a low affinity, so a high Km, about 0.5 to 0.8 grams per litre, in comparison to ADH of a Km about 0.05 to 0.1 grams per litre. In comparison to a chronic alcohol consumer, take the this example, who would have a low Km and high affinity. They'll need to have a higher concentration of substrate to get the maximum enzyme activities. This signifying that they have a high metabolism rate and can eliminate acetaldehyde more efficiently. Hence, they can tolerate alcohol better. And I forgot to mention, in reverse, for a non-alcoholic individual, the Km will be much higher, which means they've got a lower affinity. The enzyme becomes saturated only after a few drinks. For example,
Thank you, Kelly. In order for the enzyme to be fully saturated, substrate will be needed. This is an example that we plotted out in the mechanics mental plots. This shows that when a non-alcoholic consumer has the same amount of intake of alcohol as a chronic alcohol consumer, a chronic alcohol consumer would need less substrate to activate the enzyme to the maximum. Thus, that for non-alcoholic consumers, the enzyme is not activated completely, which means that the metabolism of the alcohol will then be metabolized slower when it's compared to non-alcoholic consumers. Well, I hope you enjoyed our presentation.
keep up the good marks!